Hi everyone, I am Dr. Jeeva Thapuran. Today's presentation is on cell cycle and its regulation. Importance of the cell cycle. The identification of the factors that regulate the cell cycle were discovered through the seminal work of three important scientists, Lee Hartwell, Paul Nurse, and Tim Hunt, who were awarded the Nobel Prize in Physiology or Medicine in 2001. Cell cycle. The cell cycle is a series of temporally ordered events that leads a cell to divide itself into two daughter cells. It's a complex system. The components are identified. It is a highly regulated process. Although both somatic cells and germ cells divide, they follow slightly different processes named mitosis and meiosis respectively. Stages of cell division. The cell cycle may be broadly divided into three distinct stages: interphase, mitosis, and cytokinesis. Interphase is the period between successive rounds of nuclear division, and it is distinguished by cellular growth and new synthesis of DNA. The figure shows the cell cycles in a stratified epithelium. The figure A shows light micrograph of section of a skin. The diagram B showing different type of cell cycles at the various levels of epithelium. Initially, the renewal of stem cell population by infrequent cell cycles. Then expansion of the population by rapid cell cycles. Then terminal differentiation and cessation of cell cycling, and the final stage of differentiation in skin. Characteristic of cell cycle. It consists of temporally ordered events. It cannot go backwards. Positive and negative feedback loops regulate the function of the molecules involved and ensure the coupling of the different phases. Checkpoints maintain the order of the events in case something goes wrong. When do cell numbers change? During growth and development to expand pool of cells, during tissue turnover to replace lost cells, during response to injury, add new cell to wound and to replace lost cells, during physiological changes, example lactation, infection, hypoxia, and exercise, when need to balance production with loss for tissue homeostasis, when need to remove unwanted excess after demand is met. Cell cycle details. Diagram A, cellular morphology and chromosome structure across the cell cycle. Diagram B, length of cell cycle phases in cultured cells. Diagram C, time scale of cell cycle phases. The four cell cycle phases are G1, S, G2 and M. Each cell born at the completion of M phase, which include mitosis. The partitioning of chromosomes and other cellular components takes place during mitosis. Cytokinesis is the division of cytoplasm. The chromosomal DNA is replicating during S phase. The remaining two phases are gaps between mitosis and S phase. The G1, the first gap phase, is the interval between the mitosis and the start of DNA replication. The G2 phase, the second gap phase, is the interval between the completion of DNA replication and mitosis. The G1 and G2 phases are vary in length and very short in early embryos. The phases of mitosis. The phases of mitosis are divided into prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, nuclear membrane breaks down, spindle fibers appear, and chromosomes condenses. During prometaphase, spindle fibers attach to chromosomes, chromosome condense further. During metaphase, chromosomes align. During anaphase, centromeres divide, sister chromatids move to opposite poles. During telophase, nuclear membrane reforms, chromosomes decondense, and spindle fibers disappear. Regulation of the cell cycle. Cell cycle is highly regulated to establish a state of balance of homeostasis between cell proliferation, cell differentiation, and cell death. Cells that temporarily or reversibly stop dividing and viewed as in a state of quiescence called G0 phase. Cells that permanently stop dividing either due to age or due to accumulated DNA damage are senescent. Cells undergo programmed cell death is apoptotic cell. The process is called apoptosis. The cell cycle. The cell cycle is divided in 4 biochemical stages and 2 morphological phases. The biochemical stages are interphase, first gap, the G1, replication of chromosomes, the S phase, second gap, the G2, and the morphological phases are mitosis and cytokinesis. Cell cycle checkpoints. Progress through the mammalian cell cycle is continuously monitored via multiple cell cycle checkpoints. In the G1 phase, damaged DNA is detected. In the S phase, incomplete replication is detected. In the G2 phase, again, damaged DNA is detected. In the M phase, improper spindle is detected. Cells will not progress through the phase of the cycle in which defects are detected. In some cases, if the damage cannot be repaired, such cells undergo programmed cell death, which is known as apoptosis. Non-dividing cells. Non-dividing cells are said to be in G0. Some non-dividing cells include neurons and skeletal muscle fibers are in G0 forever. Others, including fibroblasts, hepatocytes, and lymphocytes, are usually in G0 but can be coaxed into the cell cycle by growth factors or, in the case of lymphocytes, by antigen along with cytokines from helper T cells. Regulation of cell proliferation and growth. Cell cycle control takes place at 3 levels. External control by hormones, growth factors, and cytokines. Intracellular control. Signaling cascades coordinate nuclear and cytoplasmic response through MAP kinases, etc. Intrinsic control. Checkpoints within cycle to coordinate replication, repair, chromosome segregation, and cytokinesis. Cell proliferation is triggered by mitogens. Mitogenic stimuli can be provided by the extracellular matrix. The integrins in focal additions of extracellular matrix not only mediate additions to the extracellular matrix components, but also provide an assembly point for signaling molecules. Cell matrix contacts tend to be mitogenic, but cell-cell contacts usually are anti-mitogenic. Soluble growth factors allow the cell to respond to signals from more distant sources. The term is used loosely to refer to proteins that stimulate cell growth factors in strict sense. cell proliferation, mitogens, or cell survival factors. Mitogens regulate gene expression. Mitogen push cells through the G1 checkpoint. Mitogenic signals activated by growth factors exert their effects between the onset of the G1 phase and a point later in the G1 phase called restriction point. Retinoblastoma protein guards the G1 checkpoint. Progression through the cell cycle and DNA synthesis require the transcription factor E2F, which regulates more than 500 genes. During the G0 phase, retinoblastoma binds to E2F transcription factors. E2F binds to promoter elements upstreams of genes. Binding of E2Fs to RB prevents the transcription of genes, the products of which play a role in cell proliferation. Regulation of cell proliferation and growth. Examples of growth factors include the following: platelet-derived growth factor (PDGF), epidermal growth factor (EGF), fibroblast growth factors (FGFs), insulin-like growth factor 1 (IGF-1), erythropoietin nerve growth factor (NGF). Growth factors bind to specific cell surface receptors. Growth factors bind to specific cell surface receptors expressed on effector cells, which are generally transmembrane proteins with a growth factor binding domain or ligand binding domain and cytoplasmic protein tyrosine kinase domain, which is known as PTK. Growth factors selectively initiate signaling cascades. The binding to a receptor causes receptor dimerization or oligomerization and activation of the intracellular tyrosine kinase domain, which in turn mediate receptor transphosphorylation at specific amino acids within the cytoplasmic domain. The phosphorylated region of the receptor is then able to act as a docking site for the binding of specific transducer proteins, thus enabling protein-protein interactions. The transducer protein shown in this picture is SRC, GRB2, SHP, PLC gamma, and PA3K. Activation of the RAS, ERK, MAP kinase, and PKC signaling cascades upon growth factor binding. Growth factor signals activated at the plasma membrane as a result of ligand binding can regulate gene transcription, cell cycle progression, proliferation, differentiation, or apoptosis. Growth factor receptor signals activate signals to recruit adapter molecules such as GRB2, SHC, PLC, PI3K. They activate ERK, MAP kinase pathway, PI3-AKT pathway, and PKC pathways, which in turn activate transcription factors and regulate proliferation, differentiation, and apoptosis. Cytokine receptor signaling. Similar to growth factors, cytokines also exert their effects on cells by binding to cell surface receptors. There are multiple classes of cytokine receptors, of which many belong to the hematopoietic receptor superfamily. In contrast to growth factor receptors, cytokine receptors do not possess an intrinsic catalytic activity. Members of the family of cytosolic PTKs, called Janus kinases, are essential for linking the hematopoietic receptors with the downstream signaling and gene transcription. Cell cycle regulators. Cyclin-dependent kinase family, and cyclins regulate cell cycle transition points. Cell cycle pass through three switch-like transition restriction points, positioned at the G1 S phase boundary and at the entry and exit of M phase. Master regulators of these transition points include members of the cyclin-dependent kinase or CDK family of serine-threonine kinases and a family of proteins known as cyclins. CDKs are expressed as heterodimers comprising a protein kinase subunit and a regulatory cyclin subunit. Cell cycle mediators and their formation of complexes. Complexes of certain cyclins with specific CDKs possess enzymatic activity. Whenever necessary, cyclin-dependent kinase inhibitors can be recruited to inhibit cyclin CDK complexes. Cell cycle specific expression of cyclins and activation of CDKs. The cyclins categorized as cyclins D, E, A or B are family of cell cycle regulatory proteins. Different cyclins are expressed to regulate specific phases of the cell cycle. Cyclin concentration rise and fall throughout the cell cycle due to its synthesis and degradation. D-type cyclins, cyclin D1, D2, D3 are G1 regulators critical for progression through the restriction point. S-phase cyclins include E cyclins and cyclin A. Mitotic cyclins include cyclin B and cyclins and cyclin-dependent kinases during the cell cycle. Most cyclins are short-lived and their levels fluctuate with the stages of the cell cycle. The cyclin-dependent kinases and cyclin D, on the other hand, are present throughout the cell cycle. Only cyclin D is not controlled by the cell cycle. It is controlled by mitogens. It rises when a cell in G1 is exposed to mitogen. Tumor suppressors and checkpoints. Tumor suppressor proteins normally function to halt the cell cycle progression within G1 when the cell should not continue past the restriction point. Sometimes it is desirable for a cell to remain in G1 or enter G0 when continued growth is not needed, or undesirable, or when DNA is damaged. Mutated versions of tumor suppressor genes may encode proteins that permit cell cycle progression at inappropriate times. Cancer cells often show mutations of tumor suppressor gene. Retinoblastoma activation of transcription of S-phase genes. This tumor suppressor functions to halt a cell in the resting or G1 phase of the cell cycle. In resting, retinoblastoma protein contains fewer phosphorylated amino acid residues and it prevents entry into S phase by binding to transcription factor E2F and its binding partner DP1 or 2, which are critical for the G1S transition. In actively cycling cells, retinoblastoma progressively hyperphosphorylated as a consequence of growth factor stimulation. Hyperphosphorylated retinoblastoma protein can no longer inhibit transcription factor E2F binding to DNA. Therefore, E2F is able to bind to DNA and activate genes whose products are important for S phase. P53. This tumor suppressor protein plays a major regulatory role in G1 phase. Nuclear DNA damage results in phosphorylation, stabilization, and activation of P53. Activated p53 stimulates CKI transcription to produce a protein named P21 to halt cell cycle progression to allow for DNA repair. If the damage is irreparable, p53 triggers apoptosis. Unregulated cell cycle progression can occur in the presence of mutated versions of p53 because it is incapable of including the steps that would normally arrest the cell cycle. Monitoring for DNA damage. In the case of damage occurring, the DNA damage checkpoint sends alterations and activates signaling pathways that mediate DNA repair. If the damage is beyond repair, they induce apoptosis. The core molecules of the DNA damage checkpoint are sensor kinases and checkpoint kinases. These molecules prevent G1S and G2M phase transitions by inhibiting CDK activity through increasing the expression of CDK inhibitor protein P21 via p53 protein stabilization and or inhibiting the CDK activator. If the DNA damage is beyond repair, p53 dependent programmed cell death by apoptosis is triggered. Cell death by necrosis and apoptosis. Cell death may be accidental or programmed, initiated and executed through distinct biochemical pathways. Programmed cell death is genetically regulated and its role is to remove superfluous, damaged or mutated cells. Apoptosis. Cells deprived of survival factors activate an intracellular suicide program and die by a process programmed cell death, which is known as apoptosis. The rounding up of the cell, restriction of pseudopods, reduction of cellular and nuclear volumes, chromatin condensation and fragmentation, plasma membrane blebbing, formation of apoptotic bodies, and engulfment by resident phagocytes takes place during apoptosis. On the other hand, necrosis is an accidental and uncontrollable form of cell death that occurs after severe insult to the cell. During necrosis, an enlargement of the cell volume, swelling of the organelles and plasma membrane bursting with concomitant loss of intracellular content takes place. Apoptotic cells are engulfed by phagocytic cells, macrophages, and dendritic cells, which bind to the phosphatidylserine on the membrane surface. A macrophage internalizes and then degrades an apoptotic cell, reducing the risk of inflammation from cell death. Functions of apoptosis. Elimination of damaged cells is an important function of apoptosis. Apoptosis is used during development of the embryo. Extensive cell division and differentiation during this period often result an excess number of cells that must be removed in order for normal development to proceed and for normal function to occur. Homeostasis. In normal healthy adults, the number of cells is kept relatively constant owing to a balance between cell division and cell death. Homeostasis is a balance between cell death and cell proliferation. From a stable baseline population, gain in number takes place via proliferation, which includes cell replacement and cell growth. The loss in number takes place via cell death, which includes apoptosis and necrosis. Elimination of damaged cells is an important function of apoptosis. When damage to cellular DNA occurs and the damage cannot be repaired, apoptosis occurs. The normal p53 binds to a p53 responsive element within the gene promoter of pro-apoptotic protein BAX, triggering programmed cell death. Mutant form of p53 neither halt cell cycle nor initiate apoptosis. Cell continue to survive and divide. Initiation of apoptosis. Cellular apoptosis via formation of apoptosome. BAX. A pro-apoptotic protein is induced and inserted into the mitochondrial membrane to form a channel to allow cytochrome C to exit mitochondria. Cytochrome C in the cytoplasm triggers the formation of the apoptosome, a large protein complex that also requires ATP for its formation. To form this complex, cytoplasmic cytochrome C activates the APAF1 adapter protein that in turn activates caspase 9 of the caspase proteolytic cascade that will cleave and destroy cellular proteins and DNA in order to cause cell death by apoptosis. Caspases. Caspases are cysteine proteases with aspartate substrate specificity. They are synthesized as inactive proenzyme, referred to as procaspases. Initiator caspases, caspase 2, 8, 9, and 10. Synthesized as monomers, activated by death signals. Dimerization includes full enzymatic activity, mediate proteolytic cleavage of other caspases. Effector caspase proenzyme, expressed as preformed dimers activated by initiator caspases, execute the cell death program. Thank you for listening to this presentation.